that we should do an early switch in those patients who did not meet the 10% after 3 months or who didn't come below the 1% after 6 months, because we know that the second generation TKIs are more effective, and we have to find a good reason why not to use them as first line. And if we use imatinib first line, then at least we should not take a higher risk for those patients after these early time points. We should switch. Nevertheless, no study has prospectively shown that this really is effective, but it's very likely. The NSND trial, which is the trial that compared imatinib standard dose with the 2 nilotinib arms, had the primary endpoint of an MMR at 12 months. And you see on this slide that this primary endpoint was perfectly met. The yellow curve, 27% of patients on the imatinib arm achieved an MMR versus 51% in the 2 arms being treated with nilotinib. So it's much more effective, and this effect is constant over the first 3 years. This is recent data from ASH and EHA. But I want to show you that the PCR landmark analysis, as it has been presented in Amsterdam at EHA, has the same value as in imatinib first-line treatment. So this evaluation showed for the overall survival by BCR-ABL levels being achieved at 3 months. And again, you see only one curve who is significantly worse, which is associated with significantly worse overall survival, and this is the curve of patients who didn't make it below the 10% limit. The total number of patients who did not meet this criteria was much, much lower than in the imatinib-based arm. So in the German study, we had nearly 30% of patients who were in this bad risk group, compared to only less than 10% on first-line nilotinib. So that also shows that the nilotinib is more effective. Nevertheless, if a patient on first-line nilotinib doesn't meet that criteria, this is a risk factor and something should be changed. Similar things have been presented for the DECISION trial. So the trial which compared dasatinib versus the standard dose of imatinib. And you see here, very similar to the one that I showed you before on the left-hand side, that on dasatinib first-line treatment, only those patients have a significantly worse overall survival who did not meet the 10%. Again, this was only a very small cohort, only a rough around 10% of all patients, compared to roughly 30% in the imatinib arm. The interesting thing on this trial was that also a cytogenetic predictor was evaluated, and this was also in accordance to what we've shown before for the imatinib-based treatment in this German CML study 4, that only those patients lived shorter who did not achieve a partial cytogenetic response. So another strong argument that partial cytogenetic response criteria is equivalent with the 10% landmark on the molecular level. So that brings me to the point that the definitions and the ELN recommendations will be revised very soon. And since I'm part of this committee, there is a lot of discussion because there are 31 people whose opinion is brought together. And so this is just something that you should not take for granted, but it is clear that we will have molecular predictors from the very beginning on, and probably at the 3-month time point, the 10% BCR-ABL will be a hallmark to be taken. The partial cytogenetic response is much more stricter than the no cytogenetic response, so above 95% of Philadelphia positive metaphases. So we will have a strengthened, a more strict criteria at the 3-month time point, as well as the 6-month time point, where the 1% BCR-ABL or the CCYR will probably be a stricter criterion. For the 12-month, there will be, in addition to the criterion of CCYR, also the molecular predictor. So you see that we will have molecular predictors which are easy to determine. You know that for a molecular response assessment, you don't need bone marrow. It's much easier for the patient to do a blood sample, and yeah, the patients will love you. Just a short notice about one trial which shows us how treatment and how our aims could be in the near future. The so-called NST-CMR study, and you see that the term CMR is still very much anchored in also study names. This was a study which randomized patients after 2 years on imatinib treatment who did not achieve a CMR4 yet. And they were randomized to switch then to nilotinib or to continue imatinib. So the concept was maybe with implementing this would be the natural course on imatinib. By including or introducing nilotinib, you might get a deeper response, and this hope or this concept was proven. As you can see here, the MR4 rates were significantly higher, 48 versus 25% in the imatinib arm. The MR4.5 rates also significantly higher, as you can see here. So probably we might bring more and more patients by this approach to a deeper molecular response, and thereby we might bring them to a point where we could discontinue treatment. The NST first trial, I referred to that a little bit earlier, with the primary endpoint of an MR4 within Europe, showed preliminary results at the last ASH meeting, and you see the blue curve are the rates of an MMR, cumulative incidence curve. So 50% achieved an MMR after this short period of time of 6 months. And if you remember the curves that, from the NST-ND trial, the nilotinib first 300 milligram BID arm achieved the 50% after 1 year. So it seems that we can do even faster now with more experience in nilotinib treatment. So the aim probably will be that we get more and more patients to take part in discontinuation trials. The French study showed us, the STIM study, which means Stop Imatinib, 100 patients took part in that trial, and imatinib was continued if those patients had a long-term, very deep molecular response. You see here that more than 50% of these patients lost this deep molecular response within the first 6 months or so, but 40% without treatment rested in that very deep molecular response. So the outlook is that we expect that more and more patients under the more effective second-generation TKIs as first-line treatment will achieve these complete or very deep molecular responses, which might be the justification for treatment discontinuation trials. In Europe, we just started with a larger discontinuation trial, which is called the EuroSki, Europe Stop Kinase Inhibitor. We implement also patients who want to stop treatment after having achieved a very deep molecular response on second-generation TKIs. So I'd like to conclude that we find early and deep responses, and those predict for progression-free and overall survival in any of the TKIs that you can use. The second-generation TKIs, nevertheless, are associated with an earlier and deeper responses than imatinib. These early responses can be determined by both cytogenetics and molecular monitoring, as I showed you. But the molecular monitoring is more sensitive, and it allows an early detection of potential relapse or non-compliance, which also is a problem, which is uncontrollable otherwise. It can be performed using peripheral blood instead of bone marrow. It's becoming more and more important since we have more potent drugs, and the molecular endpoints of studies are needed to fulfill and to be measured. And it allows a consideration of treatment interruptions in selected cases of very deep and long-lasting responses, but this should be done within clinical trials only. And, of course, the ELN recommendations need revision concerning the impact of deeper and earlier responses. So this was my last slide. Thank you very much.